For every major scientific breakthrough, there was a time before it. A time when the world was one way, and then a time when it was different. A time when the sun revolved around the earth. A time when all bacterial illness was untreatable. A time when dots and dashes wrote our hellos and goodbyes. A time when all computers were the size of suburban homes. And for all those times, before and after, historians were both witnesses and recorders. Chemists and engineers, physicists and geneticists. Scientists from every discipline mixed a desire to discover and create, with a willingness to question the status quo. Each made the decision to push forward, spurring some of history's greatest innovations. The Science History Institute is the organization dedicated to collecting and sharing the scientific achievements that have shaped our lives. We document and commemorate the history of chemistry, chemical engineering, and the life sciences, telling the stories of innovators and inventions, experiments and analysis, pioneers and their crowning achievements, researching and preserving the past so that we may inspire a deeper understanding of the world we live in and what the future may hold.
Good afternoon. I'm David Cole, President and CEO of the Science History Institute. It is my great pleasure to welcome you to the 2021 Heritage Day Awards ceremony, which we're thrilled to present virtually after postponing last year's event due to the pandemic. Now in its 17th year, Heritage Day celebrates exceptional achievements in chemistry, chemical engineering, the life sciences, and allied fields and industries. Today, we honor the path-breaking accomplishments of three individuals: Professor Dame Carol Robinson, Dr. Peter J. Stang, and Steven Holland. We also recognize the scientists, epidemiologists, public health officials and workers, and companies who redirected their energy and talents toward fighting the novel coronavirus. These dedicated innovators and professionals have spent a year identifying the molecular identity of the coronavirus, understanding how it spreads and who is most at risk, and pursuing the rapid invention of tests, drugs, and vaccines to tackle it. In spite of setbacks and notwithstanding the many challenges that still need addressing, some of the most amazing work that has ever been done in science and public health has been accomplished over the past year. Central to the Science History Institute's mission is the collection, preservation, and sharing of these remarkable, often untold stories about the authentic, lived experiences of scientists and the very nature of scientific inquiry. The Institute captures the role of imagination and curiosity, experimentation and collaboration, tenacity and determination, risk-taking, and openness to learning from failure, as well as successes. The Institute's award-winning museum, where I'm standing today, is one important venue through which we present these stories of the innovations and discoveries that shape our lives. But these live presentations of scientific content are complemented by public educational programs, fellowships, and our much-lauded Distillations series of podcasts and articles, a growing body of digital content which is reaching an ever-expanding global audience. To accomplish this important work, we rely upon the generosity of many people. Today, I'd like to warmly thank the individuals, companies, and groups who have partnered with us on this year's Heritage Day. The Bolte Family Foundation and Francis X. Bolte, Board President, Thermo Fisher Scientific and Dan P. Shine, Senior Vice President and President of Analytical Instruments, Pfizer Incorporated and Susan Anderson, their Chief of Global Operations and Global Product Development, Waters Corporation and Dr. Lance Nicolason, Senior Director, Mass Spectrometry Business and Field Marketing Operations, and West Pharmaceutical Services and their CEO, Eric M. Green. It is also my pleasure to acknowledge two organizations with long-standing affiliations with the Science History Institute: the American Society of Mass Spectrometry and their board president, Dr. Susan Richardson, and the American Institute of Chemical Engineers and June Wispelway, their executive director and chief executive officer. And now, without further ado, it is my great pleasure to introduce Francis X. Bolte, who will present our first award, the Richard J. Bolte Sr. Award for Supporting Industries.
Thank you, David, for that kind introduction. Our father, Richard J. Bolte Sr., started Philadelphia-based BDP International with just a typewriter and a $1,200 loan in 1966. 55 years later, BDP is one of the largest privately owned logistic companies in North America, but at heart is still very much a family business, supporting customers in chemical, life sciences, and healthcare industries and humanitarian aid sector with a safe movement of essential products and goods across the world. Our father was also an avid supporter of the Science History Institute, as am I and my brothers. Our eldest, Brother Rich, is currently the chair of the Institute's board of directors. To honor our father's business and philanthropic legacy, the Institute established the Richard J. Bolte Sr. Award for Supporting Industries. Since 2007, the award has been presented annually to courageous, visionary leaders who have catalyzed growth, innovations, sustainability in the chemical and life sciences community. Among past honorees are David and Alice Schwartz, co-founders of Biorad Laboratories, Satoshi Horaba, chairman of the Horaba Limited, the Japanese manufacturer of precision instruments for measurement and analysis, and Frederick Frank, the first investment banker to specialize in biotech, pharmaceutical, and healthcare sectors. On behalf of the Science History Institute and the Bolte family, it is my honor to present this year's Richard J. Bolte Sr. Award for Supporting Industries to Steven Holland, the recently retired CEO of Brenntag AG. Steven has had an impressive career of more than 30 years in chemical manufacturing and distribution. He began as an industrial chemist in the field of water treatment and surface coatings and joined Brenntag after his acquisition of Albion Chemicals in 2006. A year later, Steven was appointed to Brenntag Board of Directors as Chief Executive of the company's European business. He was named Chief Operating Officer for the group in 2009 and CEO in 2011. During his 14 years at Brenntag, Steven has significantly contributed to the company's journey from private to public ownership and its expansion into truly global chemical distribution company and market leader, operating in 73 countries with 16,000 employees. It's also worth noting that in 2013, Brenntag became one of the first companies in the chemical sector to join the UN Global Compact, a worldwide initiative based on a CEO commitments to protect and implement universal sustainability. Under Steven's leadership, environmental protection and the responsible care of natural resources are values firmly embedded into today's company's processes, products, and governance. Congratulations, Steven, on this richly deserved award, which is given in appreciation for your vision, passion, and outstanding corporate leadership.
Well, hello, everybody. It goes without saying, I wish I could be with you, and I really do wish I could be with you today, but for reasons we're all pretty familiar with, it's not been possible to travel to join you today. But so before I go on about my award, if I could just say a quick congratulations to David Carl Robinson and to Professor Peter Stang for their gold medal awards and what an amazing contribution they've made to science and chemistry. So this is an amazing award. It does recognise my period as the chief executive of Brenntag and taking that business maybe over a decade from what was quite a regional business into literally a worldwide chemical distribution business with sales of over $17 billion. So a huge step forward, very challenging, but honestly extremely rewarding. And I had a ball a great career. In fact, many people would say it was one which lots of people would have envied. And I certainly I had a time at Brenntag which surpassed many. What I would say, though, during that process, I did have some advice from an old chairman of mine when I asked him, what can I do to be better as the CEO? And he gave me some very simple advice. And the simple advice was never stop saying thank you, and always seek to give recognition to those people who have got success in your organization, because their success will become your success. And that's something I try to do. And in actual fact, today's award is a demonstration of that. So I'd just like to say thank you so much for this award. It really means an enormous amount to me. And have a great day. Thank you.
The Science History Institute is deeply indebted to our 35 affiliated organizations, institutional partners who collaborate with us to preserve the history of science and technology and related industries. The American Institute of Chemists is one of our esteemed affiliates, and since 2003, we have jointly presented the AIC Gold Medal at Heritage Day. I am delighted to introduce Dr. David Minuta, AIC's board president, who will present this year's AIC Gold Medal to Dr. Peter J. Stang.
The American Institute of Chemists Gold Medal is our highest award and recognizes extraordinary accomplishments in chemistry and chemical engineering in the U.S. The award has been bestowed upon 74 remarkable men and women since 1926, including Nobel laureates Glenn T. Seaborg, Herbert C. Brown, Elizabeth Blackburn, and Dudley Hirschbach, to name but just a few, and luminaries such as Chad A. Merkin, Mary Low Good, Stephen J. Lippard, Jacqueline Barton, and George M. Whitesides. This year's distinguished recipient of the AIC Gold Medal is Dr. Peter J. Stang, the David P. Garton Chair and Distinguished Professor of Chemistry at the University of Utah. A life and career as remarkable as Peter's can't be summed up in just a few words. So we want to share this short video highlighting how Peter has changed the science landscape through his groundbreaking research in organic supramolecular chemistry, his inspired teaching and mentorship of students and fellows from around the world, and his exceptional service to the field, most notably during his transformational 19-year tenure as editor-in-chief of the Journal of the American Chemical Society, affectionately called JACS.
I started out in chemistry when I was an 8th grader in Hungary, and in those days I could go to a drugstore and buy all the chemicals I wanted. And ever since then I've been hooked by chemistry.
Stang and his family were forced to flee Hungary for the U.S. in 1956, yet his love of chemistry would not be deterred.
By January 1957, which was my second year in high school, I was in Chicago. I did not speak English, but I was number 1 in class in science and mathematics. I went to Berkeley and became interested in the work of Professor Andrew Streitwieser, who was then my mentor, and I became interested in physical organic chemistry.
These small organic molecules continued to fascinate Stang, now from a new home at the University of Utah.
I went to give a seminar, and one of the colleagues during the question said, "Listen, these are 90 degrees. Have you thought of making a square?" And then I started thinking. I said, now where else could one make shapes like squares, hexagons, pentagons, or even three-dimensional molecules? And that's how I became interested in self-assembly. And it's just wonderful to rationally design how you can make precursor molecules, which you then just mix at room temperature. And within a few minutes, they just self-assemble into these very complicated, very interesting three-dimensional molecules.
With equal exuberance, Stang inspires his students by visiting Utah's outdoor laboratories.
Experimentation is visual. Indirectly, when I take my graduate students to Bryce Canyon, I can show them the incredible beauty of the state of Utah. And then I say, imagine that architecture there. Now go back in the lab and use this imagination to make new molecular architectures, three-dimensional molecules. These compounds have very interesting biomedical properties, and so we have results on mice that they are quite effective anti-tumor agents. Now, obviously, a lot more work has to be done before one of these might actually become a drug.
Stang has received honors from around the world and at home.
The 2010 National Medal of Science to Peter J. Stang.
That is the miracle, the beauty of this country, where a refugee kid who did not speak English can subsequently become the editor of the flagship journal of the American Chemical Society.
As editor-in-chief, Stang would review more than 200,000 manuscripts, while serving as a committed researcher, professor, and mentor.
I am nearly 80 years old, and once in a great while I say to myself, geez, why didn't you get in a new area? But there is still an awful lot to do in self-assembly, making ever more complex molecules. And I will now remain in this area for as long as I can have young people who bring new ideas and carry them out.
It is now my distinct honor as president of the AIC board to present the gold medal to Peter J. Stang. Congratulations, Peter.
Thank you, Dr. Minuta, for the kind introduction. I'm greatly humbled and honored to receive this distinguished and coveted recognition from the American Institute of Chemists, and to join the many preeminent scientists who preceded me in receiving this recognition. I would like to express my appreciation to the selection committee for choosing me for this very nice gold medal. I am most thankful and grateful to my family for their support throughout the many years of late-night work, as well as absences due to my professional travels. I am also grateful to the University of Utah and my colleagues in the chemistry department for providing an environment conducive for first-class research and teaching, and for appreciating excellence. This award belongs as much to my just over 100 very international graduate students, postdoctoral fellows, and visiting scholars as it does to me. For even the most original innovative ideas remain just that, ideas without capable, dedicated co-workers to carry them to fruition. I would like to conclude by expressing my appreciation to the country of my choice, the United States of America, for giving me the opportunity to go from a young teenage refugee escaping Hungary in 1956 to the American Institute of Chemists medalists in 2021. Thank you very much.
And now, let us turn to the final honor we will bestow in this program. The Othmer Gold Medal is the Science History Institute's highest award and is named after Donald Othmer, a noted chemical engineer, inventor, philanthropist, professor, and co-editor of the Kirk-Othmer Encyclopedia of Chemical Technology. And just like the medal's namesake, past winners, scientists and innovators such as Sangeeta Bhatia, Joshua Boger, Yuan-Se Lee, Gordon Moore, and Robert Langer have all made outstanding contributions to not just one, but in many areas of science, technology, and industry. Joining this illustrious group of trailblazers is Professor Dame Carol Robinson, the Dr. Lees Professor of Chemistry at the University of Oxford, and the first director of the new Kavli Institute for Nanoscience Discovery. Dame Carol will be speaking today about her current work in a short talk followed by Q&A. But first, I would like to introduce Dr. Ian Milcreast, Vice President of Research and Development in the Analytical Instruments Group at Thermo Fisher Scientific. Dr. Milcreast will tell us more about Dame Carol's unconventional, barrier-breaking rise to the top echelons of science. Dr. Milcreast.
Thank you, David. Dame Carol's groundbreaking research and career, which we honor today, has always been characterized by her extraordinary ability to look beyond the limitation of what's currently known, to question what could possibly be, and to push the technology to gain new insights into 3D structure and protein function and their complexes. It's been described as one of imagination and persistence in the face of established opinion. Over a quarter century ago, Dame Carol defied early scientific opinion concerning the theorized limits of mass spectrometry for the analysis of proteins in the gas phase. Posing and designing her own instrumentation configuration and experiments, she recorded the first mass spectra of macromolecular complexes, including molecular chaperones and intact ribosomes, and laid the foundations of mass spectrometry in structural biology, and really pioneering the area of native mass spectrometry. Dame Carol's research opened a new phase of gas phase structural biology, provided insight into a range of otherwise intractable biological assemblies. Her innovative methods of broad applicability in drug discovery and contributed to characterizing both new protein drugs and understanding drug mechanisms. Dame Carol continues pioneering the use of mass spectrometry as an analytical tool in chemical biology research, more recently championing a new native omics concept to understand what specific ligands are bound to membrane proteins, which is key to understanding structure and stability, regulation of protein function, and drug action. Over the last several years, our development team has had the pleasure of collaborating with Dame Carol, her research group at Oxford, and her colleagues at Othmer Therapeutics, a biotechnology company she founded. The challenge before us was to develop a new platform capabilities that can not only analyze intact membrane protein complexes, but clearly identify attached ligands such as lipids and drugs in a single experiment, which had never been done before. The instrument that was developed was an ultra-high resolution Orbitrap tribrid mass spectrometer, which is the next evolution of the future understanding of protein function and further testament to Dame Carol's extraordinary scientific vision, brilliant leadership, and fierce dedication to passion for her work. There's so much more I could say about Dame Carol's formidable accomplishments, but instead let's look at a short video describing her high-impact research, her non-traditional career path from leaving school at 16 to becoming the first female professor of chemistry in Oxford and Cambridge, and an internationally admired, much honored scientist.
In terms of glass ceilings, I think I would like to emphasize that Carol is the very first female Dr. Lees professor that we've had in Oxford.
I actually found it quite a scary moment. I remember an email going around saying, oh, and Carol makes history today, and it was intimidated by that because I thought I've got to do a really good job or they'll never have another one. It's an experiment after 400 years. If I don't do well, then it won't be seen as a good one.
I would also say that she's very modest and unassuming. It would be incredibly easy to get an overinflated ego given the list of her achievements. Although she is Dame Carol Robinson, or Professor Dame Carol Robinson, actually when you meet her, she's just Carol.
She's really a pioneer. I mean, she was the one that opened the field of native mass spectrometry. It wasn't there before her because people used to use mass spectrometry for analyzing small compounds, for analyzing peptides, but she was the one that had the idea that it could be used for studying protein complexes.
If you are doing something that's a bit different, you have to expect that people won't always believe you or think it's a good idea, and I had quite a lot of that in my early career. There's a certain amount of sort of doggedness is required to do that. And I always say to people, if you have a conviction and you've done the right controls and you know you're on the right track, then you should keep going against the dogma.
I come from a very strong mass spectrometry background, and I have to admit that early on, while reading her papers, listening to her presentations at conferences and meetings, my initial reactions were rather skeptical. But she presented very strong experimental data, very convincing data. My research has really transitioned into studies of proteins, protein complexes, and my entry into that field was motivated in part by some of the early work that I read from Carol Robinson's group.
Once a problem is manifested in biology, Carol is very good in looking at that and kind of saying, okay, how can I actually solve this problem using mass spectrometry or related techniques, rather than simply saying, I do have a mass spec, then let's go and find a way how to employ it. And what this actually done in turn is not only open up new avenues for native MS for applications, but in a way have kind of made us think about what other new methods we need to develop.
OMAS came out of my lab based on some discoveries that we'd had about particular kind of receptor, G protein coupled receptors. This was very important for pharma. And initially we were just collaborating with pharma, and that was growing very nicely. And then we realized actually we could have our own company.
I've been actually very lucky that I did my PhD with Carol. And then later on, I worked in OMAS. So I've kind of had different throughout the years, I've had different interactions with her. As a PhD student, Carol was sort of motivating us to come up with novel ideas. So she had a very good way of saying no without telling you no.
I've noticed that if you say to somebody, oh, why don't you try this experiment? It's never worked in the past and people have been trying for 20 years, then they don't want to do it. But if you say, it'd be great if you could get this to work, I really believe it's going to work, and then it's not working yet, but it will work. There's a kind of positiveness about that.
I think that Carol actually was the one that prepared me to be a group leader.
In the first day that I met with Carol, I kind of saw another side of her, that she didn't ask me a single question about science in particular, but she directly asked me about, have I found a house in Oxford yet? So she directly was looking into other sides that make a great scientist as well.
So if I had to pick one word, I think I would actually like to pick several words, but the first word that comes to mind, I think, is inspirational.
So I think as a scientist, Carol is very perfectionist and excellent.
Consummate scientist.
She was always a big inspiration for me.
On behalf of the scientific community, and specifically the mass spectrometry community, I'm truly honored to present the Othmer Gold Medal to Professor Dame Carol Robinson in recognition of her many breakthroughs in the field of mass spectrometry and proteomics. Her extraordinary record of leadership in both academia and industry, and her unwavering advocacy and outspoken support for women in science.
Good evening. Or good afternoon. I'm absolutely thrilled and delighted to receive this award. It means a huge amount to me, and I accept it on behalf of all of my former group members and my mentors who've really helped me to get to this stage. I'm truly honored. I would also like to congratulate my co-awardees. I think excellent scientists. I'm honored to be a part of this group. So in my talk, I'm going to try to find a way to thank everybody in passing through my story of the developments of mass spectrometry for protein complexes and major pharmaceutical problems. And as you've already seen, I started as a technician, age 16, at Pfizer's in Sandwich. And for me, as Peter said, I was just hooked by this technique that allowed you to look at molecular structure, that you could fly these molecules through the gas phase and learn something about them. And the sheer challenge of trying to get a mass spectrum, a very practical endeavor, was something that I thoroughly enjoyed and felt a huge sense of satisfaction. Most of the week, the spectrometer would not be working, so there's a whole team of scientists keeping it together, and I got the pleasure to drive it. I'm still surprised by this. At 16, this was quite an expensive piece of kit, and I was allowed to drive it. So yeah, it was a real love affair for me from the start. And the sort of problems that we were looking at that time would have been aminoglycoside antibiotics. This was a big thing in Pfizer at that stage. And we were derivatizing these to make them volatile, because mass spectrometers would only work if you could get things into the gas phase, and these molecules weren't volatile. And then we would work out their structure. So it was one thing to record the spectrum. The other thing was the manual interpretation that you had to do, which I always thoroughly enjoyed and prided myself on doing this at great speed. So trying to assign the spectrum was something of a challenge. And nowadays, we would be able to look at the receptor or the channel in which these aminoglycosides actually act. So mass spectrometry, as Ian was saying, has come such leaps and bounds in the time that I've known it, that it's a very exciting field to be in today. As a PhD student, I'm grateful to my supervisor, my late supervisor, Dudley Williams, who took a chance on me, having not had the standard background into university. He offered me a PhD position. And at that time, I was looking at fast atom bombardment, bombarding molecules on a glycerol matrix such that they would be released into the gas phase. And Dudley's favorite molecule was vancomycin. And we were interested in the complex that it would form with the bacterial cell wall peptide D-ALA. And we looked at this complex, and unfortunately, although it bound to that peptide, so did the enantiomer, the L-ALA. So we clearly thought the gas phase is not going to reproduce interactions in any meaningful way, and sort of shelved that idea because fast atom bombardment could not really protect the structure of gas phase ions. We moved on another decade. Have a great and brilliant mentor, Chris Dobson, who always encouraged me to tackle very challenging experiments and things that were not considered the normal remit of mass spectrometry. And we started to study protein folding, and the molecules that we were looking at the time, one of them was hen egg white lysozyme. And to follow folding by mass spectrometry, you have to introduce another step. You have to start to label the protein. So we introduced deuterium, which is heavier than protons, folded the structure, and trapped within these partially folded states. And this was the first time we'd seen these intermediates. So this was a breakthrough for mass spectrometry to see a protein folding intermediate. So I was very proud of that work. If we think about that, as I mentioned in my interview, there was quite some controversy about whether or not, or some skepticism might be a better way of putting it, about whether or not these molecules could retain their folded structure. And we set out to try to prove that interactions, in fact, could be maintained if the protein molecules were treated in the correct way. And so we designed this instrument with Waters. I'm very grateful for their help. It has a high mass quadrupole. We changed some of the focusing and built this early QTOF mass spectrometer to go to much higher mass to charge ratios. And we got our first ribosome mass spectra, and this is Paul, who's now in Sweden, and we got a surprise. And this is what I like about mass spectrometry. You can always learn something new when you put something into the gas phase, and this is an unusual stalk complex. It was predicted to only have 4 copies of the red protein, and in fact turned out to have 6, which was totally unexpected. And this region is very dynamic and often not seen in crystal structures or EM, but readily captured by mass spectrometry, which doesn't distinguish between static and dynamic molecules, which is one of its great attributes. But of course, that doesn't necessarily mean that it's going to have the right structure or the same structure as you think it will have in solution. And one of my favorite analogies is that if you put something up into the gas phase in a bubble and it's in the atmosphere, perhaps this gives you some sort of freedom. If you're running in the air, it's much easier than if you're constrained and trying to run in a swimming pool, for example. You may have tried this. You have a loss of freedom of movement. Whereas if you're in the atmosphere, you're running, you can throw your arms around and you can really move around. So maybe a protein in the gas phase actually has more flexibility, more movement than something in solution. And so many people thought this is never going to be a useful way of looking at complexes because they're going to unfold. They're going to express themselves in ways that we don't fully understand. And one of the ways we tried to come to terms with this was to use ion mobility mass spectrometry for protein complexes. And just very briefly, protein complexes go across in a gas-filled collision cell. They're accelerated and there's a countercurrent of gas, and you can time them. And this, of course, is very important because you can relate that timing event to their collision cross-section. And this particular complex, TRAP11mer, was ideal for these types of experiments because it had either a ring or a ball, and of course the ball would come through more quickly than the ring. So we were able to distinguish these 2 conformations of 2 protein complexes that have the same mass to charge. So this, again, for me, was a sort of landmark moment where we started to show that protein structures actually did have some semblance to what we thought they would have in the gas phase. And one of my favorite molecules, everybody has a favorite molecule, is the V-type ATPase. And this will crop up a few times in my story, but Min Zhu, who is in Nanjing, she showed that we could take this complex out of the membrane. It's a very important complex that synthesizes all of your ATP and keeps the acidity of your cells regulated. And this has 29 subunits, some membrane, some soluble. And she showed that we could protect it and transport it into the gas phase with everything kept intact. And this was, for me, quite a remarkable spectrum, because all of these membrane parts have to keep in contact with these soluble parts and trundle through the mass spectrometer with 57 charges on. And in this direction, we lose subunits, we shed subunits if we activate. And in this direction, the whole membrane region comes out as one. So this was very exciting. We found, in fact, that the membrane region came out with a collection of lipids. And this led us down another direction, thinking about how lipids regulate protein complexes. And Bolte, who is now a professor at Yale, he showed that there was in fact a sort of sliding scale of lipid dependence. And along this end, proteins were inherently stable, and along this end, lipids were required for stability. We also noticed that these particular interfaces appeared to require lipids for stability. And this is very reminiscent of the story of the G protein coupled receptors, which you will know are really important drug targets because they control many bodily functions such as sight, touch, hearing, and of course entry into your cells. So very important signaling molecules. And there was always a question, are they lipid-mediated dimers? And we set out to answer this question, and in fact didn't answer that question, but answered a different question, which I found more interesting, because sometimes in science you set out to do something and you get taken down a completely different path, and that's always exciting for me. And these green molecules here we discovered actually mediate the interactions with a downstream signaling protein. This is one of the G proteins. It was a G mimetic that we used in these experiments, and we showed that we got enhanced signaling with these PIP2 lipids. As Ian mentioned in the introduction, we're always trying to think of a better way to do the experiments, and we were worried that the detergents that we were using might be influencing somehow the protein complexes and the lipids that we were observing. So we set about trying to find a way to take these complexes directly from membranes. And we did this via a vesicle preparation that we would form either via excretion, cell press, or osmotic shock. And then we would sonicate these vesicles initially, that was the method of choice, put them into the mass spectrometer and drive them through with quite high voltages and try to charge them up and actually almost pull them directly out of the membrane through charge. Sorry. And I'm just going to show you one example of this, and this is from E. coli membranes. And we found that there was a heterodimer of OMP, and that there were some new complexes of the BAM complex. These hadn't been seen before in detergent, suggesting that somehow the membrane was stabilizing these complexes and allowing us to see them in the mass spectrometer, so the dimer and its interaction with a molecular chaperone. So we reasoned that this could be a more general method for extracting complexes directly from their membrane following charging of the extracellular or the cytosolic regions of the protein, not the membrane embedded parts, but the parts that stick up rather like an aerial for charging and then pulling into the mass spectrometer. And we've shown this very recently. This is unpublished data now from solute carriers from mitochondria, which we've overexpressed. And what we're trying to do here is to trap the small molecules that pass through these receptors. And if we do in detergent, we get the very nice spectrum at the top. It's a dimer with two lipids. But if we look at the one directly from the membrane, from Hyping time, you can see that we have the dimer of the transporter and then 1 and 2 adducts of a small molecule caught passing through this transporter, two lipids binding simultaneously. So this spectrum to me is a very exciting hint as to where we will go next, because many of these transporters and receptors, we don't know exactly what they're doing. And if we can find out precisely what these small molecules are, we have a key to their function. And as Ian mentioned, we've benefited from a great collaboration with Thermo Fisher. This is Idi and Joe, who spent many days in San Jose factory working with a very experienced team to try to assemble this instrument to allow us to do what we're calling native omics, a combination of native mass spectrometry and omics technologies. And effectively what we're doing here is taking something that's existing in the membrane with either a lipid or a small molecule cargo, breaking it in the mass spectrometer, and then working out what's happening with its structure here. So it takes me back to my early days in mass spectrometry, because I can impress my students by trying to identify these small molecules from their fragmentation pathways, which is what I was doing when I was at Pfizer. So it's really important, because if you were to isolate or extract the small molecule, you would find hundreds of small molecules with every membrane protein. But this method allows you to take just the targeted ones that were actually bound directly to the membrane protein complex. So that's why it's particularly exciting for me. And I'm just going to show you one example, and that is of a mitochondrial transporter, TSPO. And you can see here that it binds beautifully to this lipid, but actually it doesn't fully accommodate the density that was found in the X-ray structure. So this lipid didn't look quite to be the correct one, and we realized it would be a great opportunity for our native omics approach to try to work out what it was that was actually sitting here. And by breaking this molecule in this way, we identified this particular lipid and modeled it back into the density, and you can see that it fits really nicely. Everything matches up and good connections are made between the lipid and the protein backbone. So we believe that this is going to be a very important and exciting way forward for studying small molecules that are hiding within these membrane protein complexes. And finally, one of my final examples at least, as you know, I came from a very small molecule background, and to me, I didn't think I would really see the day when we would take from a rat brain, one rat brain, 5 micrograms of a V-type ATPase from a synaptic vesicle, because I always thought it would just be too messy, too heterogeneous. We'd never get a good spectrum. But Diwu managed to get this beautiful data set. And you probably don't think this is beautiful, but it is, because it's incredibly complicated, which I like, of course, because it means that there's lots happening and lots that you can interpret here. But also there's many different fragmentation pathways. And we were looking for one particular thing. We were looking for a subunit that would give us the identity of this V-type ATPase as being from the synaptic vesicle and not the normal V-type ATPase. So we look in this region and we identified this particular subunit as opposed to a different mass, very small mass differences, but we were able to identify these proteoforms and also in a different ATPase lipid that binds in this ring. So mass spectrometry can contribute to these very beautiful structures that are coming out of cryo-EM at the moment. And of course, there's a coronavirus story. We sat and watched as this, of course, took over the world and wondered if we could deploy the tools that we've been developing for years, decades, to this problem. And I'm very grateful to my COVID response team, looking incredibly well-dressed here, Jani, Corinne, and Tarek, who were able to come in during the initial lockdown to look at some of the proteins that are involved. And I'm just going to tell you about one of them, and that's the N protein, which spontaneously cleaves into many different proteoforms that then interact either with the RNA or with cyclophilin, which is part of the immune response. And we can eliminate this with a cyclosporin A. We can also look at the dimers and how they package the RNA for efficient genome packaging. And then we can look at interactions with patient antibodies. And we've been very fortunate to receive antibodies from sera from patients who've recovered from COVID-19 infection. So these are just some of the spectra. We've also taken a look at various of the players in this receptor-mediated entry of the SARS-CoV-2 virus. So that's very much work that's ongoing. And with that, I'd like to give you a very quick summary timeline. I started my research career after a career break in 1992 looking at protein folding intermediates, and then molecular chaperones, which we put through new instrumentation to try to maintain. Started to look at peripheral membrane proteins and ring proteins, demonstrating different shapes of complexes. First membrane proteins in 2008, that was a big milestone for me. Some very nice work with Art Laganowski, which I didn't have time to show today, looking at how we could find specific lipids that stabilize membrane proteins. My favorite molecule, the ATPase. GPCRs I've put here because they were so difficult for us to do because they were so floppy, and trying to maintain their interactions was a real challenge. And then, of course, the virus, which is ongoing and extremely challenging. As mentioned, I'm delighted and honoured to be the first director of this new institute in Oxford. It's a brand new environment for innovation through discovery. For the first time in my life, I'm going to have a lab not in the basement. I'm on the second floor with views and daylight, so this is a big breakthrough. And we're mandated to have breakthroughs at the convergence of these different scientific disciplines. Currently, we've got 8 departments coming in, and the building is open within a month or so. So this is an exciting time. We're just about to decommission and move all the mass spectrometers into their new home. But my main goal with this institute is to try to create something that I would have loved to have worked in at the start of my career, and that will then be my legacy and my challenge for the future. And with that, I would like to thank all my former group members, and there are too many to put on one slide now, but I've decided to thank and acknowledge all of them. But particularly, I'm proud of all of the ones who've gone into industry, but also have their own academic groups. And all of the people who have their own academic groups are now shown here. And these are all over the world, which is wonderful. I look in and check on how things are going. And when travel was permitted, I tried to go to everybody's lab once they'd got set up and to visit. So I'm inordinately proud of all of their achievements and watch from afar. I also would like to thank the societies, the American Society for Mass Spectrometry. I was discussing the other day that I always felt very welcome there, even though my work was sometimes considered controversial. I always felt that I was welcomed and supported in that environment, and similarly with the British Mass Spectrometry Society. And I'm grateful to the ACS and the Royal Society of Chemistry, who've also supported me throughout my career. And finally, I would like to thank my research group, current, past, and future. This is our away day, our annual away day, and of course we haven't been able to have one this year, so this is the year before. We go away to a college and talk about science, and then go and have a nice meal somewhere and try to get a photograph of everybody together. So unfortunately, this is the previous year. I had great plans for the lockdown one, but those will have to wait. And I'm of course grateful to everybody who's supported and funded our research over the years. And I thank you very much for your attention, and I'm very happy to answer any questions that you have. Oh, I would just like to show you the very nice medal that I received. Unfortunately, I can't wear it because it doesn't have a hook, but it looks great in the box, and I'm so proud. And thank you again to everybody who made this possible, and to my family who have to put up with me many times when they would rather I didn't work so hard. So thank you all.
Well, Dame Carol, many thanks to you for the excellent talk, and really, I would say, for honoring all of us by accepting this honor today. So thank you for that. To echo your words, your research has really taken us all down many interesting paths, and we're most grateful for that. And I've no doubt that your talk has engendered many interesting questions. Doubtless members of our audience will want to ask those. And to moderate that discussion, we have with us today Dr. Vicki Wysocki, who is professor and Ohio Eminent Scholar in the Department of Chemistry and Biochemistry at The Ohio State University. Dr. Wasaki.
Thank you, David. I would like to add my congratulations to all of the winners today, and especially Carol with the Othmer Medal. I'd like to start, there are several questions that have come in, and I'd like to start with one about your decision to become a chemist. What led to you deciding to become a chemist, and were there any particularly inspirational people who helped with that decision?
Well, that's a great question. Thank you. I think almost everybody would say it was their teacher, and I'm no exception. I was at an all-girls school, not particularly interested in many things, but our chemistry teacher really inspired me. He was just made it so exciting. I remember taking on problems that I didn't even have to do. You know, I was that interested in it that I would read up voluntarily at home and get really into the subject. So I wrote to him recently and told him what he'd started, and he remembered, so that was good.
Yeah, that's great. Was the position at Pfizer, the fact that it was mass spectrometry, was that just by chance? Or when you interviewed, did you know you would be using a mass spectrometer?
That's a great question too. Actually, I interviewed and they told me about mass spectrometry, but I was only 16, so I didn't know what it was. I remember they asked me a question. They said, oh, well, if it's heavier, will it go further? Or if it's lighter, would it go further? And I remember I just thought, well, I don't really know. I don't know how it works. So I just guessed. I think I got it right. So that was lucky. But yeah, no, I had no clue. And I also tried some other techniques. I tried NMR and chromatography as well, but I just loved the mass spectrometer. So I was very fortunate, I think.
I feel very fortunate too to have landed on mass spectrometry early on when a teacher in undergraduate school gave some special lectures on mass spectrometry. I think we couldn't have imagined all the changes that would have occurred in mass spec over all these years. You mentioned doggedness being necessary. I would say you also show a great boldness. You published sometimes spectra that maybe some other people would have hesitated to publish. And so I'm just curious, you mentioned having great controls. So I guess I wanted to ask, was it really boldness or was it really just strong science?
Well, sometimes I look back and think, oh, maybe we pushed that a bit far, you know, maybe that wasn't right. But in general, I don't think anything was wrong. I think sometimes we just pushed it to a new level because we were excited by it. So is it doggedness or boldness? A bit of both, I think. And I also think I don't really enjoy writing something that's too boring or safe. I want to push a bit and I want to push boundaries. And if with that, you do have to suffer a lot of rejections and a lot of pushback, but I still think it's worth it because it does move the field forward. And it does, you know, if people are critical, you can always think of more experiments to prove that you were correct, or new techniques come to the fore, and then you can answer the questions in a different way.
I think you're absolutely correct that when you pushed things, it pushed the field forward and so many people followed up. So it's been really, really great for the field. I have a question about the COVID year that we've just been through. I know that you usually travel a tremendous amount. What has it been like for you to be at home for this year? And has there been any special benefits, unexpected benefits of being home this year?
That's an interesting question. I don't miss hanging around in airports particularly. I do think flying was an opportunity to write and to think about things, so it wasn't all time wasted. I miss my colleagues hugely. I miss chatting. I seem to have frozen. I miss chatting with people about science a lot, and I don't think it's quite the same to do that on a Zoom call or any other platform.
I agree with you on that. Some in-person interaction is really helpful. I know that sometimes when you have gone and given talks or interviews, people have perhaps asked to not mention your 8-year career break to have your children and get them up to a certain age. Although there's much more of child rearing than just, oops, we seem to have lost Carol.
Hello, everyone. My name is Alexis Pedrick. I'm the I'm the Director of Public Engagement here at the Science History Institute. I just wanted to let you all know we're having a little bit of technical difficulties getting Dame Carol back on the stream here, but we are on the phone with her, and as soon as we can get her connected, we will be bringing her back in live for the chat. It looks like I can see her coming back in now, so I'm going to say goodbye on the stream here, and we'll bring Dame Carol back on in just a moment.
Hi, apologies. I don't know what happened at all. Yeah, it was.
We're back, so yeah. It's good. I was starting to ask a question about your career break, and I think it would have multiple parts, and you can sort of answer in any order you want. One part is, how difficult was it for you to go back after an 8-year break? And then what do you say to people who say, well, we shouldn't mention it because you're really exceptional, and we couldn't let other people think that they could do this because really it wouldn't have worked for them. It would have only worked for somebody as special as Carol Robbins.
Yeah, well, certainly I don't believe that for one moment. I think I was very fortunate and I very much enjoyed it. I don't ever tell people that that's what they should do or try to recommend it, because I think the world is changing, and it's easier to stay in touch now than it was when I was taking my career break, when there was no internet breaking and doing all these things. There was just the library. So I really felt I couldn't do the job that I wanted to do if I was at home. So I made a decision to take the time out, and I don't regret it. I found it very hard coming back, very hard. I put myself on an IT course, but it didn't prepare me for the challenges of where mass spectrometry had gone during my career break. So I worked quite hard, I think, to catch that back up again. I read a lot and tried to work out what had been going on in the 8 years that I'd missed. And I do know that sometimes I would get into trouble for publishing something and not necessarily citing the right person because I'd missed what had happened in the field, even though I'd tried to research it. So my history was missing for that period, but I think I've got that sorted now. But it was quite difficult. And also, I think the biggest thing was I lost a lot of confidence during that period. And I think before I went on the career break, I was fine at giving talks. When I came back, I hated it. I was really terrified. So the confidence thing was a big thing that I had to work on.
That's interesting. I know that sometimes I work with people who have confidence issues early in their career. I hadn't thought about being off and going back after a large gap, and it could contribute to being a little more nervous about those early talks. I know a question came in that says it's a little bit of an unusual path that you've taken in the sense of knowing that you had to modify instruments. You didn't just have to do science, right? You made a choice to do some science where the instruments didn't yet exist that could do that science. And so you had to know something about the technical side of the instrument and develop that while also developing the biology or chemistry or whatever I should call it. And so what advantages is the question? Retrospectively, what advantages have you gathered by getting to know the instrument side of things?
Well, that's a great question. And I often say that I wasn't afraid of it because when I was at Pfizer, almost every day we were taking it apart and pulling it to pieces and putting it back again. And so I was never scared of slightly ruining it. I think people are a bit scared of that sometimes. They say, oh no, we've just spent all this money, we can't touch it. And I say, oh no, we've got to touch it, it's not quite right. And we've got to take it apart and see why it's not working in the way that we want it to work. And I do remember when I had my first research instrument that I'd bought myself, and I said, oh, the pressure's not right. I want to drill a hole in it. And they were just horrified. So, don't you dare drill a hole in it. But they then did actually effectively drill a hole and put a valve there for me. So I think having that very early introduction of it's not working, you better keep fixing it, was actually quite useful for me.
I think that's actually great. And I know that I have some young people sometimes who say, well, how could I possibly be involved in instrument development? I don't have an engineering background. I don't have an electronics background. But they have to come to understand that if you understand the chemistry or the biology that you want to do, you can imagine what you want the instrument to do, and other people can help with some of those parts. We have a very specific question on what you see as the potential for mass spec in vaccine research and development.
That's a great question. There's a lot of work on antibodies and antigen binding, and I think that is going to be a great area for mass spectrometry. It already is. Lots of people are working on that. I'm actually very happy to see that a lot of the COVID-19 research has involved mass spectrometry, including looking at complexes. So that's a very exciting thing. And I haven't myself done any vaccine work, but I know that there's lots of people who are trying to do these experiments.
We also have a question about native messy membranes. So native messy membranes from sonication or clean, somewhat artificial nanodiscs or micelles. What's in the future? Do these approaches have a role to play in native mass spec of membrane proteins?
All three of those, I think, will have great roles to play in the future. I do think there's some very nice work with nanodiscs and SMALPs and lipodiscs, all of these things. I quite like messy membranes because I think they kind of bring you closer to the truth of what's in the membrane, and you don't influence it by your selection of how you treat it. Of course, it's difficult, and of course, it has to motivate new instrumentation, but I think that's kind of why I like it, because it's very complicated. and challenging. I like things that are challenging.
You like things that are challenging, and so I will go ahead and insert a question about that that has to do not just with scientific questions that are challenging, but you also have a lot of opportunities to take on this directorship or to become an editor, associate editor for JACS, and so on. How do you choose among all the different opportunities that are presented to you? How did you decide which professional opportunities to take on?
Yeah, that's a great question. I often think about this, and sometimes I think there are things that you do to sort of pay things back a bit. For example, the Royal Society of Chemistry sponsored my degree, and I'm eternally grateful that I got that degree, otherwise I'd still be a technician. So I decided to be the president for 2 years because I thought this is my payback time to sort of return that and to make sure that other people could still benefit from those routes. And with the JACS editorship, associate assistant, whatever, I think it's associate assistant, I think it's a sort of service to the community of mass spectrometry, because it's always been a great journal for mass spectrometry, and I'd like to see that continue. So I took that role for that reason. I sometimes, I think, take on too many roles, but the directorship of the Kavli, I couldn't really refuse, because I really am passionate about interdisciplinary science. I trained as a chemist, but I spend most of my time in biology now, and I'm also passionate about understanding more physics. So this is a great opportunity for me, I think.
Thank you. I also wanted to ask some questions about your relationship with companies, both mass spec companies or biopharma. How has that played a role in your career?
Yeah, that's also been very important, because when I was young, I, would approach companies and say, "Oh, this is what I'd like to do," and they'd say, "Oh, there's not a market for that, we don't want to do it." And I'm sure you've had this too, Vicky. And you have to sort of keep going. But I really want to try it. I really want to see if it will work, and then maybe other people will find it interesting, and it will become a market. So I think there was a bit of maybe doggedness again, that I'm not going to give up, and if you won't help me, I'm going to try and modify it myself. So it's probably better if you help me, otherwise it probably won't work. So I think I did keep going, and I do remember people saying, "I don't think that type of instrument's going to work." And I just said, "Well, I'm going to build that one, otherwise I'm not going to buy one." So I think you have to sometimes be quite dogged, but in general, I think people have been very supportive, and with so that was early days with Waters, and more recently with Thermo. They will say for a while, "Oh, that can't really be done," but then eventually it can be done, I think. We're still working on some of the things, but often it's a matter of time, and you have to be patient, and I'm not particularly patient, but sometimes things don't work yet, but they will.
I'm also curious to know what you think of the role of mass photometry in conjunction with native mass spectrometry. Do you see that playing a role as we continue down this path?
Yeah, absolutely. I'm very proud to be associated with mass spectrometry, because it's part of the vision for the Kavli, and two great scientists that I mentor as well. So I'm delighted that they've formed this company and are launching their equipment all over the world. And the combination is very powerful. So in solution and in the gas phase, you have these complementary methods. And we've been using it as well now for membrane proteins. So it's very exciting, and I'm delighted to see that breakthrough, yes.
We also have a question about the number of female biotech CEOs and founders. That remains low. And as a founder of OMAS, do you have thoughts on how to improve this imbalance and bias in the industry in general? And I know that's not an easy question, but maybe some thoughts.
No, I mean, throughout my career, people have said, oh, particularly in Oxford, they seem very keen for people to have companies. And I just remember saying, oh, no, I've got too much to do already. I can't possibly do that. You know, my priorities in my research. And then I realized that it actually could work very well with research. It allows you to explore other angles. So, for example, with the GPCRs, we would never screen many molecules for activity, but having a company allows you to go down that path. And I think it also changes slightly your questions, so you can think about things differently. The fact that not many women do this is quite interesting and something that we need to sort of work on. And one of the reasons I think is it's quite intimidating initially to pitch your idea in front of sort of all male investment panels, and that is changing, but only very slowly. So most of my pitching was to all male investment panels, and I did find that quite nerve-wracking. I have a wonderful female CEO in my companies who's doing a great job. So I'm very pleased that we've managed to get to that stage, and I go in and just admire all the things that are happening, and I think it runs very well with having a research career. So I hope that more women will try this path.
Maybe related to that and down a different branch, I know you're very proud of all of your former students and postdocs who have gone into industry or academia. You had the slide where you showed the ones who have academic careers, and you have quite a large number of those. I was wondering if you have any insight on how you have been so inspirational to have so many former trainees go into the academic route.
Well, I'm not sure I can take credit for all their wonderful successes. I guess I've always tried to encourage different thought and to do something different. I always think there's no point in doing something that's the same as everybody else is doing. So maybe it's trying to be original, because I think if you do something that everybody else is doing, you don't really get noticed or make a name for yourself in any way. So trying to encourage people to think differently, perhaps, is something I would like to think I've done.
I know that you are very hands-on with your group members. I saw that on a visit to Cambridge, in fact. So I was really impressed by how much conversation you have with each group member, and you're very inspirational to them. What else did I want to ask? We have some additional questions. One on innovations in medical research. So what innovations in medical research do you think might be possible in the future as more advances in mass spectrometry technology come about over the next few years?
That's a great question. I'm kind of interested in looking more closely at tissues and receptors within tissues. And of course, this isn't really quite possible yet, but it's not so far removed. We've started, of course, with the mitochondrial experiments, and the tissues are just one further stretch of that type of experiment. So we've also been looking at things from liver, from other organs. I would like to think that we could inform drug discovery in a much more meaningful way. So, for example, with receptor dimerization of EGFR, for example, if we could see that a patient had this particular phenotype, then we could treat more knowledgeably, rather than just assuming that they've got these mutations. So maybe they have a dimer or they have a monomer. If we could actually read about the state of their receptor, then we could prescribe better. So that's kind of an ultimate goal, but it's quite distant, I would suggest, at the moment.
but still seems possible in our lifetime.
I hope so. I really hope so, because then this area of research would have massive medical implications, I feel.
And you know, the native mass spec field, I always still think of it as a new field, but it's actually getting quite mature. It's been you started doing native mass spectrometry a long time ago. Yeah. I wonder if you still encounter skeptics. Do you still encounter skeptics who think that, you know, structure in the gas phase, is that really something we should be looking at? I know I still encounter skeptics. I'm curious how often you still encounter skeptics.
Yes, I do, especially if I go against something that other people believe, particularly that's, oh, it's just rubbish, it's just in the gas phase. So yeah, there's still times when you have to defend that corner, but less so, I would say. And you know, you've helped tremendously in the US in waving the flag for native mass spectrometry and in helping it get accepted. So, and all of the people that go off around the world and set up these different labs and groups, I like to think that that's also a sign that it's becoming accepted in a lot of institutions.
It is, and I think part of it is just educating.
Yeah.
People. You know, there are places where it doesn't exist yet, and where the faculty may not have heard of it, or people in a company may not have heard of it, and so they are naturally skeptical when they first see it. But then if you provide enough data and enough comparisons to established techniques, they start to see the value and start to think of new experiments. So it's been really fun, and I know you see that all the time. Are there things that I haven't asked you about that you were hoping you might get a question about?
I can't think of anything. Okay.
Maybe what advice do you have for young people as they are choosing whether to enter into the mass spectrometry field? Would you tell them it's still, you know, I encounter people sometimes who say, wow, it's had this great rise, but surely it's starting to plateau. Do you think mass spec is at a plateau, or do you think it's still on an upward steep slope?
I think it's very much on an ascendancy, personally. I remember when I finished my PhD, my supervisor, Dudley Williams, said, everything's been done in mass spectrometry, you know, you should think about something else. And that was before electrospray, of course, and that really transformed everything. But I see a lot of new developments on the horizon, which I think will make yet further transformative discoveries possible. So I think it's a great career. And actually, I was talking with my friends in ASMS recently about how women have been well accepted and well viewed within the American Society for Mass Spectrometry. There's never been any problem with women's acceptance by this society. So I do think young people would have a great career in mass spectrometry.
Well, I'm told it's time to wrap up the questions. I want to thank you, Carol, for answering all these questions. It was really, really informative and fun. Congratulations again on the award, and we'll go back to David now.
Thank you.
Yeah, I second that, Dr. Wysocki. My thanks to both of you for a very stimulating discussion on a very thought-provoking talk. And I want to congratulate the audience for asking such excellent questions and seeding a really good discussion. And this discussion concludes our Heritage Day programming, but it has been a wonderful day. I want to thank again all of our honorees. It is our honor to have you with us today and to recognize your work. Our presenters, of course, our sponsors, without whom none of this would be possible, and all of you for attending what has been a virtual, but I would say a truly global event today. Thank you for being here. You will see on your screen, I think in a moment, ways that you can, there it is, ways that you can engage with the Science History Institute, support our work by texting us or by visiting our website. We appreciate that. That allows us to keep the good programs and the good things coming. And we look forward to having you stay in touch and to joining us at other events and programs we have coming up in the year ahead. Again, many thanks for being here, and congratulations once again to our honorees, and look forward to doing this again next year. Thank you all for coming.